Hello and welcome to Science Research Weekly, Season 4, Episode 29, Proving Einstein Wrong. Get ready for the power of iNaturalist in biodiversity research, mechanisms behind lightning strikes, multi-region brain organoids, a programmable chromosome engineering platform, an idealized double-slit experiment, why hardware may need a new foundation, animated maps in R, and a grant from PepsiCo on novel proteins. This episode covers materials from August 1st to August 7th. Let's get started with our research articles. First on the docket, iNaturalist accelerates biodiversity research. Researchers conducted a systematic review of iNaturalist data and found its usage in peer-reviewed research has grown by a factor of 10 in just 5 years, with data ranging from 128 copies and 638 taxonomic families. The idea of citizen science, people taking pictures of biodiversity, has really helped science. Next up, photoelectric effect in air explains lightning initiation and terrestrial gamma ray flashes. So exactly what triggers a lightning strike? Researchers now think that lightning is initiated by photoelectric feedback, where essentially high energy electrons cause an avalanche that keeps amplifying to other electrons spreading out. This also helps explain X-rays and radio waves emitted from lightning, which apparently lightning does. And these are known as terrestrial gamma ray flashes. So despite humans seeing lightning for millennia, we're only just starting to really figure out some of the nuts and bolts of what causes lightning. All right, let's switch over to a fascinating and slightly worrisome sector in the article Multi-Region Brain Organoids Integrating Cerebral, Mid-Hindbrain, and Endothelial Systems. So researchers managed to create a multi-region brain organoid from human cells that integrate cerebral, mid-hindbrain, and complex endothelial organoids into one structure. So a bunch of mini organoids sort of modeling the different parts of the brain into one single one. Now, certainly a step forward to better modeling brain function and help understanding how the brain works to help correct issues in human health, but I'm not sure about the ethics of going too much further. Like, I mean, what's the dividing line between creating a brain organoid and a brain? I mean, certainly there's a large gap, but I think we need to be careful. Speaking of worrisome sectors, here's another paper, Iterative recombinase technologies for efficient and precise genome engineering across kilobase to megabase scales. So researchers are able to edit DNA on the chromosome level with a technique called programmable chromosome engineering, which allows for, as it suggests, large-scale DNA manipulations with scar-free editing. Again, very interesting stuff, but, I think this care needs to be taken. Human therapeutics is on the table as a list of applications for this technology. And look, if something goes wrong while flipping huge segments of DNA, what goes wrong will probably go really wrong. So I think we certainly need to be careful about how this technology is applied. All right, enough pontificating. Let's switch over to inhibition of the minor spliceosome restricts the growth of a broad spectrum of cancers. Very, very fascinating. So this is small-scale changes in cell biology, talking about the minor spliceosome. So minor splicing helps with the correct gene expression of only about 700 human genes, but it's important for those. So if you were able to disrupt minor splicing, you can disrupt things downstream. And it turns out that disrupting some of the mechanisms of minor splicing can suppress tumors while sparing healthy cells. So some cancer cell lines need minor splicing more than regular cells, and so if you can target that specific ones, you can really go to town. So really fascinating kind of piece of small-scale cell biology. All right, speaking of small, let's turn to single atoms with the article Coherent and Incoherent Light Scattering by Single Atom Wave Packets. So from what I understand with the experiment, which I can only see the abstract, but leaning heavily on the Science Daily summary with sort of the provocative title of Einstein was wrong, or proving Einstein was wrong. So scientists actually performed an idealized version of the double-slit experiment with single atoms and single photons and found that under These circumstances, light indeed cannot be observed as a wave and a particle at the same time. I think Einstein thought you'd be able to, if you looked at it precisely enough, you'd be able to parse them out, but turns out that's not the case at the most ideal situation. And I certainly don't blame Einstein. Sort of outcomes of quantum mechanics, the idea of wave particle duality, it's really not intuitive. So there we go. All right, closing out our article time here with just a short article from Advanced Science News called Surfactants Steers Supraparticle Structure. A bit of a primer on how to modify supraparticles. So a supraparticle turns out to be a cluster of smaller building blocks like nanoparticles that can display collective behavior. So you have a bunch of nanoparticles, and depending on how you adjust it, like with things like pH and temperature, etc., you can have all sorts of different sort of formulations of spheres that maybe be smooth or crystalline or rough or even like collapse and buckle and down upon themselves. So pretty interesting piece of material science. All right, let's turn our attention to some research tools. Volume 113 or 113 of the Journal of Statistical Software. More articles from that volume are now available, including Dameflame, a package Python providing fast interpretable matching for causal inference. Box HED 2.0, scalable boosting of dynamic survival analysis. PyMLE, a Python package for maximum likelihood estimation and simulation of stochastic differential equations. There is estimating spatial dynamic panel data models with unobserved common factors in stata. And statistical processing monitoring.jl, a general framework for statistical process monitoring in Julia. Speaking of Julia, interesting article on Julia Hub by Anshul Singhvi about how maybe hardware needs a new foundation. The problem, like legacy tools don't really allow for agile workflows or be able to combine like the benefits of high fidelity with scale, whereas something like Dyad is able to branch that, and that might be a solution toward bridging the gap. Now, also, there's some really good discussion about how AI won't simply solve all issues in engineering, that some non-machine learning approaches are still needed and will be needed for the relevant future. All right, over on the Do Loop with Rick Wicklund. He is in a blog post, 3 ways to estimate parameters while fitting a distribution to data. So it's the method of moments, the maximum likelihood optimization, and solving for the root of a complicated function. And it can be accomplished using the Gumbel distribution. Then there are 2 R-bloggers posts. First from Chris Bowden, Introduction to Generalized Linear Models with Prussian Horse Kicks. And then second from the Jumping Rivers blog, Animating Maps with ggplot2 and gganimate. There are no new CRAN packages of note, so we'll end our time here with some research funding. A single grant here, actually surprisingly from PepsiCo, on novel proteins, peptides, and amino acids with health benefits. That's what they're looking for, and they definitely want things to be novel, things that haven't been done before. And unsurprisingly, they want it to have little to no taste or change in texture in a powder and liquid beverage application. And it needs to have human relevant data just demonstrating health effects. And it needs to be safe and a clear route of commercial sales. So say PepsiCo wants these proteins, peptides, amino acids with health benefits for use in their products. Supraparticle Colotastic. Thanks for joining me for Science Research Weekly. Science on.